Cellular development is a turbulent physical process. Living organisms are local, low-entropy anomalies, forced to constantly burn energy to fight off thermodynamic decay. Because the developing brain is a dissipative system, it is continually subjected to unavoidable thermodynamic noise. And here lies a core biological paradox. The adult brain wires itself into a hyper-precise specific circuit, despite being built in a highly stochastic environment. This animation shows a standard misconception, assuming neural wiring depends on a deterministic mechanism. In this model, every neuron requires a precise pre-programmed address code to find its target. But in the noisy reality of a living system, these rigid addresses fail. Developmental algorithms actually require noise to build precise structures. It is a functional necessity for biological organization. The brain operates as a self-organizing system, utilizing chaos to achieve a level of complexity that a rigid, pre-programmed machine couldn't sustain. Evolved systems frequently utilize random processes as core inputs for complex algorithms. The immune system provides a clear proof of concept for how stochastic biology operates. The biological challenge is straightforward. The body must identify and fight an infinite array of unknown, randomly evolving external pathogens. This diagram illustrates the immune algorithm solution. It generates billions of unique T-cell surface receptors entirely at random. A deterministic, pre-programmed response cannot anticipate a completely novel pathogen. A massively randomized pool of receptors ensures that at least one will physically match the invading shape. This randomization phase is a mandatory step in the immune system's developmental program. Algorithmic growth incorporates noisy, non-deterministic assembly to achieve outcomes that rigid programming cannot. In neurobiology, we see a similar mechanism with DSCAM, a cell surface protein found in Drosophila that acts as a neural identification tag. This animation demonstrates the sheer scale of DSCAM's molecular variation. A single gene splices algorithmically to produce nearly 40,000 distinct protein variants. These proteins exhibit strict homophilic binding. Variant A can only lock together with another variant A. It completely ignores the other 39,999 variations. The biological fact that creates a paradox is this. Every single neuron expresses a randomly selected variant of this DSCAM protein. If the purpose of a molecular ID tag is to find specific targets, assigning that tag at random defies logic. Consider a massive database. An ID number only functions if a central authority logs it and knows who needs it. Under a deterministic wiring model, a random ID acts like a secret password that no one else knows. It is useless for matchmaking. Statistically, if neuron A receives a random DSCAM tag, neuron B cannot predict that specific tag to form a matched connection. DSCAM cannot function as a target address. Its massive randomization serves a completely different structural purpose. The paradox resolves when we shift the perspective. The random DSCAM ID is not designed for target neurons to read. It is designed strictly for the cell to define its own self. This diagram visualizes the math of the random assignment. Choosing 1 out of 40,000 variants virtually guarantees that no two neighboring neurons will ever share the same ID. Neurons expand their dendritic trees through stochastic branching. The process is noisy and unpredictable, much like an apple tree branching chaotically to reach sunlight. As a neuron branches into dense space, its tendrils inevitably collide with its own branches. This animation shows the self-avoidance mechanism. When branches of the exact same neuron touch, identical DSCAM tags bind, triggering a localized repulsion signal. Macroscopically, this repulsion forces branches to grow outward and away from each other, maximizing the volume the dendritic tree covers. Without the initial noise of chaotic branching, the neuron would not need a self-avoidance mechanism to manage its shape in the first place. The algorithm actively harnesses two layers of noise, random protein assignment and chaotic physical branching, to efficiently spread the neural network through space. If DSCAM dictates spreading and self-repulsion, we must establish how specific synaptic connections actually form. Neurons possess a baseline biological drive to form synapses at all costs, regardless of perfect accuracy. The phenomenon of the autopsy provides evidence of this drive in its most extreme form. When researchers culture neurons in total in vitro isolation, the growing cells have no correct partners available to connect with. This illustration shows what happens next. An isolated, desperate neuron, will physically curl inward and form functional synapses onto its own body. Spreading out through DSCAM repulsion prevents this pathological self-synapsing. By expanding outward, the neuron puts itself in contact with others. The DSCAM mechanism is a permissive instruction. It establishes the spatial geometry so that local instructive chemical cues can execute the final matchmaking. Biological specificity is never a single blueprint. It is a composite built from multiple overlapping mechanisms. Stepping back from single cellular mechanisms reveals the vast, interconnected reality of the developed neural network. This diagram maps the sequence of development. Random ID generation enables self-repulsion, which structures the noisy physical branching, which ultimately delivers the neuron to its local targets. This shows that the inherent stochasticity of biological growth is actually a resource for the system. A purely deterministic view of the brain is biologically inaccurate and mathematically insufficient to sustain complex life. Forcing absolute deterministic precision onto every single synapse would require a genome larger than the organism itself just to store the data. Utilizing stochasticity offloads the computational burden from the genome directly into the physical environment. The brain's architecture is achieved because it does not suppress the chaos of the universe. True biological precision is guided noise.